In this video, we're going to talk about polysaccharides. Recall that polysaccharides contain 3 or more monosaccharides joined together. So every single monosaccharide here is going to be joined by a glycosidic bond. These glycosidic bonds can be in either the alpha or the beta orientation. So just like we discussed with disaccharides, polysaccharides are basically more monomers or more monosaccharides joined together, but they still have an orientation in their bond. The types of glycosidic linkages that these polysaccharides will have can dictate many of its properties. So we're going to discuss the polysaccharides involved in energy storage, and we're going to see that they will contain mostly alpha glycosidic bonds. For the structural components, or the polysaccharides that have structural roles, we're going to see that they have beta glycosidic bonds. And these types of bonds is what's going to lead to a three-dimensional structure that will facilitate its role or its biological function. So here's an outline of the types of polysaccharides we are going to be discussing in this video. So here, polysaccharides up top, we're dividing that into 2 categories based on their functions. There's going to be the energy storage function and the structural role. We're going to talk about each one of those functions in both plants and animals. So let's start with plants. We're going to talk about first the starch polysaccharide, which is composed of both amylose and amylopectin polysaccharides. Starch is the main carbohydrate found in the seeds and roots of plants. So that's going to mean corn, potatoes, and pasta. We are going to find starch in these foods. Starch is the way in which plants store their energy. So when they have excess carbohydrates that they're not going to use, they can go ahead and store them in these polysaccharides that we call starch. So there are 2 forms of starch. There's the amylose and the amylopectin. Amylose will make about 20% of starch molecules, while amylopectin makes the other 80%. All right, so let's look at amylose and amylopectin structure a little bit more closely. First, we need to start by defining the types of glycosidic bonds that exist in both amylose and amylopectin. So what we'll see is that they are polymers, meaning they are long chains of repeating glucose units. So each one of these monosaccharides is a glucose monosaccharide. They are going to be joined by specifically a 1,4-alpha glycosidic bond. And sometimes we call this the backbone, because in amylose, that bonding pattern is going to exist. It's going to exist just as a linear chain. That's in contrast with amylopectin, where we're going to have that same backbone, meaning we still have repeating glucose units bonded by a 1,4-alpha glycosidic bond. But in addition to that, amylopectin is going to have a branching point, where the carbon in the 6th position is going to form a bond with the anomeric carbon of another monosaccharide. So what this means is that the difference between amylose and amylopectin is that amylose, we call this unbranched. There are no branching points in amylose. Whereas in amylopectin, yes, it contains the same 1,4-alpha glycosidic bonds as amylose, but it also contains these 1,6-alpha glycosidic bonds, which would be this one here, where you have the carbon 6 of one monosaccharide and carbon 1 of another monosaccharide. And here it would be the alpha link. So this here is 1,6. Notice how in amylose, the 6 carbon does not have any glycosidic bonds in that position. So let's see what this means in terms of its structure. So amylose again is unbranched, while amylopectin is branched. So we can see the depictions here, where each one of these hexagons is a glucose unit. And then you'll see that in amylose, because of these particular bonds that it has, because they're in the alpha position, this is actually going to result in the chain adopting this helical structure. We're going to see that for both amylose and amylopectin, since they have the same backbone. With amylopectin, we can depict branching points as it's depicted here. So this is a branching point, and that's where the 1,6 glycosidic linkage is. And so the difference, again, is just this branching. Now, the fact that they form these helical structures, this allows them to be water soluble, because through that helical structure, the OH groups are not buried within the structure. That means that they are able to hydrogen bond with water. That is going to make them water soluble. Additionally, the human digestive system does have the necessary enzymes to digest this polysaccharide. That enzyme is called amylase enzymes. And they will carry out the hydrolysis reactions of these polysaccharides, similar to what we discussed when we discussed the hydrolysis of disaccharides. So how this hydrolysis works is this enzyme will be able to remove the glucose units at the very ends, and it can do so sequentially until it gets all of the glucose units that it needs for energy. Okay, so when that happens, when the amylase enzyme cuts off a glucose unit, the glucose is then absorbed by animals, and then it can be used for energy or it can be stored as glycogen. And this is now where the energy storage for animals comes in. So we've talked about amylose, which was the unbranched version of starch, amylopectin, which is the branched version of starch. And now animals will consume plants and they will consume starch, and the amylase enzyme will chew it up to get those glucose units that now can either be used for energy immediately or it can be stored as glycogen. Glycogen is going to be a polysaccharide. So let's look at the energy storage molecule in animals. The glycogen is the major form in which carbohydrates are stored. We do not store carbohydrates as monosaccharides nor as disaccharides. We store them in these glycogen polysaccharides. They are similar to amylopectin. Glycogen, again, is a polymer of repeating glucose units. So this is how we're going to store those glucose monosaccharides in a polymer. They're linked in the same way, 1,4 and 1,6 alpha bonds. But the difference between glycogen and amylopectin is that glycogen will just have more branching than amylopectin. But they are otherwise very similar. They're going to have that same helical structure in the backbone as shown here. This is the helical structure. And then it's going to have more branching than amylopectin. So glycogen, again, is this large polysaccharide all filled with these glucose units that can then later be used for energy. They are mainly stored in the liver and in muscle cells. So when glucose is needed quickly for energy, like when you're exercising, for example, these glucose units will be hydrolyzed. So they will be cut off like that at the ends, and they can continue to do that until you get all of the glucose that you need. And then those glucose units, or those monosaccharides, after they are cleaved, they're going to be further metabolized to release energy in a process termed glycolysis, which we are going to talk about in the unit of metabolism. All right, so we have looked at all of the polysaccharides involved in energy storage for plants. We looked at that for animals in the form of glycogen. And again, what all of these had in common is that they all contain these alpha glycosidic bonds. Now let's move on to talking about the structural polysaccharides. These are going to have beta glycosidic bonds. Let's start with looking at plants. So in plants, we are first going to look at this molecule here, which is called cellulose. Cellulose is found in the cell walls of pretty much all plants, and this is what gives plants that support and rigidity. In example, wood, plant stems, and grass. So cellulose is also a polymer of repeating glucose units, so that is how it's similar to the other energy storage molecules we just talked about. But the difference is that it contains a 1,4 beta glycosidic bond rather than the alpha that we talked about. So these beta glycosidic linkages, the impact of this is that they end up creating these long linear chains. And the fact that they make these long linear chains because of the direction of this bond, that is what allows them to stack in sheets. And they're going to make very extensive three-dimensional arrays because of this bond. So let's look at that a little bit more closely. So here what this image is showing is this is one linear polymer, meaning there is no branching. That is one linear polymer of repeating glucose units, which is cellulose. And as these polymers stack together, they form these very fibrous structures. So cellulose polymers will stack in sheets, and the stacking is mediated by hydrogen bonding. And now, since those OH groups, those hydroxyl groups that exist on all of these monosaccharides, since those are buried within the structure, so you would have hydrogen bonding here in between the structure, for example, because they are buried in there and they are already hydrogen bonding to each other to form these sheets, these polysaccharides turn out to be insoluble because those OH groups that would normally be available on a monosaccharide to hydrogen bond with water, now they are hydrogen bonding with each other to form these sheets, and therefore they are not available to hydrogen bond with water. this is going to make them insoluble. Now, beta glycoside bonds are cleaved by an enzyme called beta glycosidase. This enzyme is present in cattle, so other ruminant animals, and that enzyme is present in the gut bacteria in these animals, and that is what allows them to be able to digest these cellulose polymers, so they will digest it in the same way, cleaving the glycosidic bond and grabbing that glucose unit. But it turns out that humans don't have that bacteria in the gut. This is why we cannot digest cellulose. So while cattle and others can grab nutritional benefit from eating grass and leaves, humans cannot, because we don't have the enzyme that is going to cleave that glycosidic bond in order to extract that glucose unit. Instead, cellulose is just going to pass through the digestive system in humans, and this is what we call insoluble fiber in our diet. And this is going to be important for gut health. So even though we cannot derive nutritional benefit, meaning we cannot derive calories from this, we cannot absorb it because we cannot cleave it, it still passes through our gut, and it is important just for the health of the bacteria and the environment in the gut. Okay, so we have talked about cellulose in plants, and we talked about how this is going to be important for the structure of plants. It's present in the cell wall. And we talked about how consuming them as animals, that we as humans specifically, we do not gain calories from this insoluble fiber, but we do gain benefits in gut health. All right, so now let's turn to the polysaccharides that have structural roles in animals. These are going to be what we call for short GAGs, but they are called glycosaminoglycans. So glycosaminoglycans, also known as GAGs, are a group of unbranched. They are unbranched carbohydrate derivatives. So what we mean by derivatives is that you are going to have OH groups in your monosaccharide units replaced with some other functional group. So in this case, in the case of hyaluronate, this one is going to be different. It replaces an OH group. So that's an amine group there, or an amide group there. This is going to be a carboxylate group. So as you look through, you're going to see that there are a lot of OH groups that have been replaced. And this is what we mean by carbohydrate derivatives. Now, these derivatives, you're going to see some of them have a beta glycosidic bond. This is an alpha glycosidic bond. Okay? But these types of unbranched carbohydrate derivatives, they're going to be very important for connective tissue and joints, because their structures are going to be gel-like. They form this gel-like matrix, and it's going to act as a lubricant in these joints and connective tissue. All right. So here is a summary of all of the polysaccharides we talked about. Specifically, importantly, I should say, energy storage molecules are going to have these isoglycosidic bonds. Specifically, amylose, which is unbranched, has the 1,4, while amylopectin, it has that 1,4, but it also has those branching points, which happen in 1,6 alpha glycosidic bonds. The structural components in plants, we talked about cellulose, which is a major component in cell wall, and it's what gives plants their rigidity and their support. Lastly, we talked about glycosaminoglycans, which are important in connective tissue and joints in animals. And then very briefly also, we talked about glycogen, which is stored in liver and muscles, in order to provide fast energy when needed by using an enzyme that cleaves off those glucose units and then can turn glucose into energy. So in the next unit in metabolism, we're going to talk about how these glucose units can be used in order to generate energy. That is going to be called glycolysis. And we're going to go through that series of reactions in that part of the course.